just a little background on biliary tract cancers. So biliary tract cancers are cancers of the bile ducts. This is a tumor type that really management of biliary tract cancers hasn't significantly changed in the last 10 years or so. The mainstay of treatment is cytotoxic chemotherapy, and the median survival remains in a range of about a year for patients with advanced stage disease. And so the background is we really wanted to investigate a novel therapy for this cancer using immunotherapy. There have been a number of trials of immunotherapy in biliary tract cancers. Those trials have been somewhat disappointing, with response rates that have averaged in a range of about 5 to 10%. And so in this trial, we wanted to see if we could improve upon that, combining a PD-L1 inhibitor, which is atezolizumab, in combination with a MEK inhibitor. MEK inhibitors seem to have some single-agent activity in biliary tract cancer, and there's also some preclinical evidence that the combination of a MEK inhibitor and a PD-L1 inhibitor may be at least additive and potentially synergistic. And so we thought that it was worth studying this combination versus atezolizumab monotherapy. So this was a randomized phase 2 trial. Patients with advanced unresectable biliary tract cancers were eligible. They had to have had at least one and not more than 2 prior therapies in the metastatic setting. And they were randomized 1-to-1 to atezolizumab monotherapy or the combination of cobimetinib, which is a MEK inhibitor, at a dose of 60 milligrams daily, plus atezolizumab. And they could continue therapy until progression or intolerance. Patients were stratified in terms of randomization according to disease site, and the primary objective was progression-free survival. So this was a positive study in that we met our primary objective. So we enrolled a total of 86 subjects, of whom 77 received at least 1 dose of study therapy. They were randomized 39 patients to atezolizumab monotherapy and 38 patients to the combination of cobimetinib and atezolizumab. And the primary analysis was progression-free survival. And at the time of analysis, the progression-free survival was almost doubled with the combination versus atezolizumab monotherapy. So 111 days for the combination and 57 days for atezolizumab monotherapy. So that was statistically significant, and so we were pleased with the result. That being said, response rates were low in both treatment arms. So the progression-free survival benefit was driven by more stable disease in the combination arm than atezolizumab monotherapy. There was only 1 objective response in each treatment arm, so response rates were less than 5% in both treatment arms. Adverse events we thought were manageable and were consistent with the known adverse events of the study drugs. This is a combination that has been studied in multiple tumor types, including colorectal cancer and melanoma, and the adverse events were similar to what have been seen in those tumor types. So as compared to the atezolizumab monotherapy, the combination, we did see more CPK elevation. We saw more nausea, vomiting, and diarrhea, and more thrombocytopenia and rash. Aside from that, the adverse events were quite balanced between the 2 treatment arms and were tolerable and manageable. A little bit of background here. MEK inhibitors are thought to be immunomodulatory. So there's a lot of preclinical evidence that MEK inhibitors can modulate the tumor microenvironment, promoting CD8 T cell infiltration. And the exact mechanism for why this is happening is somewhat unclear. MEK inhibitors seem to have effects both on tumor cells as well as direct effects on lymphocytes. So we were lucky to get some paired serial biopsies from patients at baseline and on treatment. And so that enabled us to really compare the effects of the combination versus the effect of atezolizumab monotherapy, so to directly isolate the additive effect of MEK inhibitors on the tumor microenvironment. The number of paired biopsies was limited, but in our initial analysis, what we found was that although there was not an increase in CD8 T cell infiltration in the combination, we did find an increase in CD8 to FOXP3 ratio. So FOXP3s, of course, are markers of T regulatory cells. And so this initial evidence does suggest that MEK inhibitors were modulating the tumor microenvironment, promoting anti-tumor immune responses. I think the main conclusion is that this was a positive study and that the combination of atezolizumab and cobimetinib met its primary endpoint and significantly prolonged progression-free survival. That being said, although this is an interesting combination, and I think it certainly warrants further investigation in biliary tract cancer, the low response rates that we're seeing in both treatment arms definitely highlight the challenge of immunotherapy in this tumor type. And I think that really this study is potentially a basis for other studies. I really wonder whether an additional agent could potentially be added to this combination to make it even more efficacious. And potentially, if there are other immunotherapies, this could be a new treatment to compare those immunotherapies to. I don't think that this study is, I think the results of the study do warrant additional confirmation. This was a relatively small signal finding study. And although we're pleased with the end result, we're pleased that we met the primary endpoint, certainly additional research is needed in this tumor type. So we've been in some discussions with the sponsor of this study and the drug company that was generous enough to provide these two agents to us, which is Genentech Roche, about next steps. I think that the results certainly suggest that there is a signal here that needs to be further investigated, and we hope to have some news about what's going to happen next in the near future. I think that this was really an amazing effort, this study. This is a relatively rare tumor type, and to get the number of patients that we did in such a short period of time is fairly remarkable. So I just want to acknowledge all the different study sites that participated in this study, the various study investigators, CTEP, which is an arm of the National Cancer Institute in the US, and then, of course, the patients and their families.